This video from Chapter 4 is going to be covering the various diagnostic techniques that we use to figure out what bacteria is in something, on something, in the air, that sort of thing. There are basically 5 techniques that are involved in identifying bacteria. One thing that we typically do is try to actually grow the bacteria so that we can examine it. There are some techniques that we'll talk about later where we don't have to do that, but in general this is what's typically done in microbiology. The first step is we inoculate something, and that's basically meaning we add the microorganisms. The second step is to incubate. Once we've inoculated something, we can't see the bacteria because they're so small. But if we put them into an incubator at the best temperature they're going to grow at and let them grow, we can eventually see the evidence of them in something that we call a colony. So once we've got that done, depending on where our sample came from, we can have a bunch of different types of bacteria, and we may only want to look at one type. So we can pick that type off of the growth media and further separate it to make sure that we are working with one thing, because a lot of the techniques we're going to talk about, it really works better if we have just one type. Now, some of the other tests that rapid tests that we are going to talk about, we don't necessarily have to have just one type. The test will still work. But when we're looking at this, the bacteria using conventional methods, or maybe ancient techniques if you want to call them that, we really do need to be working with one. And then after that we've isolated them, then we inspect them. Say look at them closely. That could mean we're looking at what their capabilities of, can they break down red blood cells? Can they convert one compound to another? That sort of thing. And then once we've done all of that, taking all this information that we've gathered, we can end up naming them. Okay? And that's ultimately the final step. Now, when we say naming them, we don't name, oh, I like that one, I want to name it Fred, or let's name that one Myrtle Soup. Now, that's not what we're talking about. They all have names that we use, scientific names. So here's a little pictorial here of what I was just telling you. They're talking about inoculation. There are various different ways of inoculating things. If you're looking at for a virus, you may use an egg here. If you ever have a flu shot, one of the things they ask you, are you allergic to egg products? And if you are, they don't want to give you that particular vaccine, because that's how they make the vaccines for the flu virus. And then we have that next step, incubate. This is a little chamber here that has a specified temperature, keeps it constant. And we usually set that at its best, its optimal growth. And then here you can see on this plate there's some different colored ones here, and that's not what we want. So they're taking a device and they're picking up one of these colonies, and then they're going to put it onto another plate to make sure that they've got it just by itself. And then here they're showing you inspection. The lab assistant here, or lab manager, whatever, is using a microscope. We could be using a microscope or other techniques. And here, under the identification, they're showing you a bunch of different possibility of tests, biochemical tests, DNA analysis, immunological. I won't be touching on the DNA analysis in this, at least not right now. It's a little more complicated. Just suffice to know that we can use the DNA analysis to determine what something is, and it's a very good technique. So, a little more detail. Isolation. They're trying to demonstrate here that you got a sample from somewhere. Let's say you've got a sore throat, and you go to the doctor, and they send in the nurse, and she takes the cotton swab and she rips the back of your throat off, if you've ever had that done. At least that's what it feels like. And then she would put it on, or he would put it on a plate and allow it to grow. And you can see here, this was a mixed culture. They're showing you they've got these rod-shaped bacteria and then the spherical ones, which are called cocci. And they put them on the plate, and they're going to grow over time. At this point in time, they're showing you here that this is microscopic. We cannot see this. But once we've allowed them the opportunity to grow, we can actually see evidence of it. It's where and they're calling this the macroscopic view. So each one of these little bumps here is called a colony. And each one of those colonies came from one original cell. At least that's the way we look at it. So you put one cell on there, give it 24 hours to grow, and it's reproduced enough so that you can actually see it. Here's some of the standard ways that we will do the isolation for bacteria. The most commonly used one is this one that's called a streak plate. And then you take the sample and you streak it back and forth on part of the plate, and then you end up sterilizing this thing. This is called a loop. And you go back and forth, and then you do that a number of quadrants, and you can see here this has got a bunch of mixed colonies right in here, and you can't see very much. But as you get towards, this was the first streak, this is the second streak, and this right here is the third one. And you can see that we've got actually 3 different kinds of colonies that we can see easily. We got the yellow one, we got the red one, and we got this creamy off-white colored one. And so that's the standard technique that we use. If we're working with something that is more concentrated, we'll sometimes use a loop dilution, where we actually end up diluting the sample so that you can get the individual colonies here. The problem with that is you might not see something if you dilute it too much. And then if you have a sample that's not very concentrated, you can put a larger volume on a plate and use this sterile glass rod to spread it around. And you can see there that a mixture of the yellow and red ones, and you may even have that off white one there. So those are some of the isolation techniques that we use. And again, the streak plate's the one that we use the most commonly. So if you want to look at this kind of in a summary, these last few slides, we start off with the inoculate, and this is for my visual learners here. Then we incubate, give them the best temperature for growing, and then we do isolation on them if it's a mixed colony. This one looks like it's not mixed, that's good. And then we do the inspection. What's on my plate? And then we can do additional stuff and identify what the bacteria actually is and give it a name, like Fred or Myrtle soup. Okay. So the different kinds of media that we have available, we classify them to the 3 properties. And when we talk about media here in microbiology, we're not talking about social media or the news media or that kind of thing. We're talking about things that we grow these organisms on. And so we can classify them by their physical state, their chemical composition, or their functional type. And we can use, of course, all 3 of them to describe one particular media. And we'll go into that in this next little section here. So I'm showing you here a liquid that's pretty apparent what it is. This can be used, this is typically used for growing either large amounts or doing biochemical tests, which I'll talk about a little bit later. We have a semi-solid one that's it's halfway between solid and liquid, and that one's actually a couple of fairly specific tests are used for that. This one here is checking to see if the bacteria is motile. In other words, is it able to move through the environment easily? And you can see this is uninoculated. This is a tube where they've inoculated, and you can see the bacterial growth around the little stab point here, but you can still see that the media around it's somewhat clear. On this one, they stabbed it, and the bacteria was motile, so it grew all throughout there, and the media looks pretty cloudy. This one, they've actually used a growth indicator in with it, and so wherever there was growth of the bacteria, you got the color change. So that was definitely a motile one. And then you have the solid one, and it says it's reversible to liquid. The solidifying agent, we're able to melt around boiling point, and then it cools down. It solidifies more around, somewhat above body temperature. And this is what we typically use for either isolation or preservation, is the solid kind of media. So with the liquid physical state, we use the term broths or milks or infusions if you ever come across them. Um, in the lab, we use something called Luria broth, named after a person who formulated it. There's something called brain heart infusion broth, those various sorts of things like that. Uh, I've also mentioned the biochemical tests. This is a particular one that they would use to see if something is able to ferment a particular sugar. So they put a sugar in here, and the bacteria is able to use that sugar, and in the process of using it, it produces a gas, and that gas collects in this little tube here, and that tells us that not only is it utilizing the sugar source, but it's producing gas, and that tells us something about the organism. I also mentioned that we can use it for growing large volumes as well. This is probably about 1 liter worth of media in here. And if you get into industry where they use bacteria to produce things, they'll be growing it in even larger volumes. They could be a 10 liter up to 10,000 liters, that sort of thing. On the semi-solid, you take that liquid and you'd add a solidifying agent that we call agar at a concentration less than 1%. So it doesn't take very much to do it, but it does give you not as solid as a solid, but not liquid either. If it was liquid, we wouldn't be able to do this motility test because the bacteria can swim, can move around, and the liquid moves just by handling it. But on the semi-solid, the liquid doesn't move, so it's a good way of testing to see, does the bacteria move, since I know that the media does not. There's at least one other biochemical test that uses it, but typically we see this being used for motility. And then solid is our last physical state. They call for 1 to 5% agar. Typically it's 1.5%, still not very much, but it gives you a nice firm surface, and that way we can actually isolate colonies. And that's what we kind of need to do. And here they're comparing a tube of solid media with a container or a tube of liquid, and you can definitely see the difference there. All right, if we look at chemical content, there can be media that we call synthetic, and that's where you take it and you weigh out absolutely every component so you know exactly what's in it. You weigh all these, you need this many amino acids, that much of these amino acids, that much of this. We add these other ingredients, everything that it possibly needs, so it's chemically defined. A complex media, we are not 100% sure what is in it exactly. If you take something like a sheep's red blood cells and put it in the agar, which we do, if you get a sheep from one field, several miles away, you take blood from sheep there, and it could be different, just based on what they're eating, that sort of thing. So complex, we don't know, but it's really good for growing things because it's what we consider to be pretty rich. And then I'm going to stop and pick up on functional media on the next one.